Yes, so this is the third video on Clostridium perfringens where we will be discussing about the Nagler reaction. So this is one of the identifying reaction where you will get to understand that how toxins can be neutralized by antitoxins. So in this Nagler reaction, as you get to see, the medium which is used comprises 6% agar, 5% peptic digest of sheep blood, 20% human serum and 5% egg yolk with neomycin sulfate. So this is basically the composition of that particular medium. Now when you are, this is a plate, right? And when this is basically being used for, or this particular test is being done, you see that one part of the plate is having the antitoxin or antitoxin, that will neutralize the toxin, whereas other part of the plate is having the toxins, right? And it is not seeded with antitoxin. Now what happens? Let's take that this is a toxin which is used, that is lecithin. Now this lecithin, in presence of this alpha toxin, lecithinase C will cause the formation of phosphorylcholine. You know, I have already told that how this lecithinase C basically disrupts the membrane. So this is basically the toxin which is produced. Now what happens? Now this is a plate which is having an antitoxin. In this part there is no antitoxin over there, and antitoxin is being seeded over here. So antitoxin will neutralize the toxin and the toxin is removed. And as a result, what is happening? There was no precipitate of any kind of cell debris or cell death, any kind of thing. So there is no formation, any kind of precipitate. So you get to see that it is written no opacity. So no opacity, that is there is no any kind of opaque precipitate which is being deposited on this plate. But on the other hand of this plate, you get to see that there is antitoxin was not there. So toxin is there. So this reaction has happened, and since this reaction has happened, you have got to have the deposits of what is called as the deposits of the cells, and which is your, that is why it will generate your opacity. So you will get opaque patches onto that of the plate. So here we get to understand that what is the role of antitoxin, and this antitoxin can be used for vaccine development. This antitoxin can be used in a very earlier stage of this particular disease, where toxin can be directly neutralized by injection or passive incorporation of antitoxin. So that is basically your Nagler's reaction. Other minor toxin, as we have talked about in the last video, so that is your gamma and eta toxin, which are minor lethal. It is not that much lethal. Then comes the delta toxin, which is lethal and which is hemolytic as well. Then is the theta toxin, which is basically supplemented with oxygen, and it is like streptolysin O, which we have discussed in the streptococcus and pneumococcus videos. Then came the kappa toxin, which is a collagenase which degrades the collagen. Then comes the lambda toxin, which is your proteinase which degrades the protein. Then is your mu toxin, which is your hyaluronidase. And last but not the least is your new toxin, that is your decarboxyribonuclease. So you understand that this microorganism is basically a galaxy of all the different toxins which are basically making the cell, synthesizing the cell. It's a building block of cell. So they are degrading slowly all the building blocks of cell. So that is why the cell is basically killed, and you have necrotizing effect on the tissues, and that is why this organism is also termed as flesh eating, or you have to have amputation or degradation of cell in specific sites. So this is all about this video, though I will again repeat this video, that is this video is having a reaction. I am talking about Nagler's reaction, in which we have taken a plate having the composition which I have already discussed. One half of the plate is having this antitoxin, another half of the plate do not have antitoxin. Now what happened? When the organism grew, the organism produced toxin, but this antitoxin, this toxin which is produced, an example was taken, that is lecithin, alpha toxin, which is lecithinase, which has resulted in the toxic effect. But because you have antitoxin on the one half of the plate, the toxin has been neutralized. So there is no deposition, no opacity on one half of the plate. But on the other half of the plate, there was no antitoxin. So there was opacity on the other half of the plate, and which suggests that antitoxin or antitoxin can neutralize the toxin and can be used as a vaccine, as a passive immunization process to save the patient. Now further, you get to see that there are some minor toxins which I have talked about, like alpha and eta, which are minorly lethal, that is not that much lethal. Then came the delta, which is lethal and hemolytic. Then comes the theta, that is O2 supplemented, and it is like streptolysin O. Then it is kappa, which is collagenase, lambda proteinase, mu hyaluronidase, and mu deoxyribonuclease. That's all about the video. Thank you.